All right, let's get started here. Chapter 12, the cell cycle. All right. All right. So again, here's your roadmap. Again, the cell cycle, right? The life cycle of a cell in its division. No matter what you're looking at, all cells basically want to divide and grow, right? So the four phases of the cell cycle we'll start with, and then how do cell division take place, and what mitosis we'll cover in detail, and cytokinesis. Okay, and sometimes these are used incorrectly. Control the cell cycle, right? And then what happens when it gets out of control, which is going to be cancer. Okay? So cells are right to the division of pre-existing cells. You remember this, the cell theory, right? All cells come from pre-existing cells, right? There are two types of cell division in us, in animals, in general, plants, in eukaryotes. There's meiosis and mitosis. Okay? Both forms of cell division are accompanied by cytokinesis. Okay? Cytokinesis, right? The cytoplasm of the cell divides into two distinct daughter cells. This is the actual process of the cell division itself. Okay, we'll touch on it a little bit later. All right, so meiosis and mitosis are usually accompanied by cytokinesis, right? The division of the cytoplasm into the two daughter cells. Mitosis, genetic material is copied, right, and divided equally between the two cells. Sorry, guys, I still have this bronchitis here. So this is just cellular replication. So the daughter cells are genetically identical to the parent cells and to each other. They're clones. Okay? Bacteria do this. Bacteria, this is how bacteria divide. They make copies of their genetic material, and they divide into two exact copies of the parent cell. Okay? That's mitosis. Okay? Asexual reproduction. Meiosis produces daughter cells that are genetically different from each other. Okay? Have half of the hereditary material as the parent cells, and we'll do that in the next chapter. Okay? That's sexual reproduction. All right, so how do cells replicate, right? Cells must replicate for life to exist, and that's really the whole point, right, of our existence is to replicate. Okay? Basic steps for cellular replication. First, you got to copy your DNA, right? You got to copy all the information, so both cells have all the information. You really have to copy all of your organelles and everything as well. You have to separate the two copies now. After you've copied your DNA, now you've got to separate the two daughter cells and dividing the cytoplasm to create two complete new cells. So you've got a cell here, it's going to copy the DNA, here's DNA, here's its copy DNA, and then it's going to eventually divide into two daughter cells that are exact copies of each other, right? So what are chromosomes? Let's get back and talk about that. A chromosome is a single long double helix of DNA wrapped around these proteins called histones. And this is how we package up our DNA. Okay? It's all wrapped up around into that double helix we've looked at before. And then it's packaged up and wrapped around these things called histones. Okay? Again, DNA encodes all the genetic information required for the cell to grow, divide, and reproduce. Now, a gene, and we'll talk about this a little bit later, is a section of DNA. Okay, so let's say you got your DNA here. This little section here will be a gene. And a gene is a section of DNA that codes for specific RNA. And since RNA then goes to protein, right, the central dogma of biology, DNA, RNA, protein, that gene codes for a specific protein. You'll have a gene for insulin. You'll have a gene for hemoglobin molecules. You'll have a gene for all of the different proteins that you use. All right, so red light, green light, the cell cycle. Some cells divide all the time, right? Cells in the surface of our skin, our epidermis, right? That cells are constantly dividing, and then you lose and replace your cells every so many days, 72 days. So those cells are constantly dividing. Mucous membranes are constantly dividing. The ones in your mouth, right? And the ones where the mucous membranes are where the inside of your body interacts with the outside, the environment, right? All the way in your respiratory tract, your gastrointestinal tract, your urogenital tract. All these mucous membranes are constantly dividing and replacing cells. Some cells divide when there's signal to divide. Okay, cells in the organs like the liver that can regenerate. Normally they don't, but if you get physical damage to your liver, it can actually regenerate. You can lose up to 80% of your liver and it will grow back. But may be triggered to divide if the organ is damaged. That's what we're talking about. You get damage to your liver, it will trigger the regeneration process. But other cells can divide with signals too. If you get a deep enough cut into your skin, those fibroblasts will start dividing and you'll start growing scar tissue, right? Your hair cells keep growing. Right? Some cells don't usually divide. Okay, cells of the nervous tissue don't divide. It's not 100% true. They're finding it now. Some of them do divide, but in general, your nervous tissue cells do not divide. Once they're mature, if you damage them, they're gone forever, right? So if you have an injury that involves nerve damage in the spine, those nerves cannot be repaired. It's not like the liver where they can regenerate. Okay? And there are a total of 4 stages in this, what we call the cell cycle, when it replicates. Okay? Interphase. An interphase consists of the G1, the S, and the G2 phases. And then you've got the M phase, which is the mitotic phase. So the dividing phase of eukaryotic cells is called mitosis. And the non-dividing phase is called that interphase, which is G1, S, and G2. All right, so look at some concepts here. We talked about this before. Cell division mitosis functions in asexual reproduction, bacteria. When we replace tissue, growth, right? When we're growing, right, our cells are going through mitosis. And wound repair, I talked about. If you get a damage, you get to grow a scar, or you replace it. So cell division distributes identical sets of chromosomes into the daughter cell. Again, it's like cloning. Okay? You're making an exact copy of the parent cell. So 2 daughter cells should be exactly like the parent cell in terms of the information they have. So these are key concepts in here. So mitosis, again, cell division used for asexual reproduction, growth, and wound repair. All the genes come from one parent cell. Right here, one parent cell is going to divide into 2 daughter cells. Okay? Meiosis, which is the next chapter, is cell division for sexual reproduction, and it's used to make the egg and the sperm. Okay? Gametes, or egg and a sperm, they're called gametes. They're also called sex cells or germline cells. You need to learn all those definitions. Okay? Somatic cells are all the rest of the cells in the body. If you're not an egg and you're not a sperm, you're a somatic cell. Okay? Ploidy number are the number of sets of chromosomes. Okay? So we get half our chromosomes from mom and half our chromosomes from dad, right? So we have 2 sets of chromosomes. Okay? So somatic cells are diploid, and that means they have 2 sets of chromosomes. That's all the cells except for a sperm and the egg. Gametes are haploid. They only have 1 set. Okay? And we'll go to that in more detail in the next chapter. So one thing that you need, that's why these are in red, mitosis preserves the ploidy number. The daughter cells have the same number of sets of chromosomes, the same number of chromosomes as the parent cell. Meiosis changes the ploidy number. It halves the ploidy number. Okay? It cuts the number of chromosomes in half. Cells go from being diploid to haploid. Okay? And you need to do that because fertilization, the joining of 2 haploid gametes, restores the diploid chromosome number. So during fertilization, a sperm and an egg fertilize to make a zygote, okay? And now the ploidy number goes back up to being diploid. And that's why you have to have the ploidy number. Because if you had 2 sets in a sperm and 2 sets in the egg, and they had fertilization, that would give you 4 sets of chromosomes. And we'll learn later on, 4 more chromosomes is not better. It's not good. Okay? All right, so chromosomes contain a single long double helix, the DNA deoxyribonucleic acid, wrapped around those histone proteins. Okay? And normally they don't look like the sex. Normally they're more diffuse. Okay? But DNA encodes the cell's genetic information. This is what people think of when they see a chromosome, though. That's why we use it. And genes in a section of DNA then encode for a specific protein or RNA. It codes for an RNA, which then goes and makes the protein, or is used to make the protein. So chromosomes can be stained and observed under the light microscope. Okay? Remember, ribosomes is where the RNA goes to make the proteins. You need the organelles. Okay. So here's some stained ones right here, paired threads I used to think of now. And here you look at this, now understood to be chromosomes, right? That's what we know of chromosomes now, these little paired looking thread things they saw when they stained cells, right? So here's an unreplicated chromosome, just one of them. And you look on here, this little piece, this one physical spot on that chromosome is a gene, okay? So now you're going to copy your DNA before you replicate. So now you've got two chromosomes. They're the same information though. So if this is chromosome number 1, these two copies would also be chromosome number 1 and chromosome number 1. Okay? And you've got that copy of gene 1 in the same spot on the new daughter chromosome, right? Now you get what's called condensed replicated chromosomes. This is when it condenses up into that sort of X shape that people think of. And there you can still see gene 1 in the same spot on this one as it is on this one, okay? And you have to do this when you want to go through division. All right, so duplicated chromosomes condense DNA now. Duplicated chromatids condense into chromosomes, condensed chromosomes. A chromatid now is one of the single strands of DNA. They are joined together in this thing called a centromere right here in the middle, right? And you've got this centromere region, and you've got these kinetochore, these microtubules, okay, right, that attach to the middle here. And we'll talk about what they do in a minute. So this whole thing is a chromosome, and it's made of two chromatids, right? So the kinetochore microtubules attached to the kinetochore. Here's the kinetochore. Okay. So this is one chromatid, and this is another chromatid. Together, they're going to be a chromosome. Now, we'll be careful to go through this. Okay, because when they start to divide, there's a couple little things you have to remember. All right, so let's go through the cell cycle now. So cell cycle is the growth and division of eukaryotic cells. Okay, the length of the cell cycle is about 24 hours to complete, and this depends on the organism, and it depends on what cells. Remember, some cells never go through division. Some cells divide faster, some cells divide slower. Okay, so that's an approximate, right? The cell cycle is divided into two main parts. Interphase, which is the non-dividing phase, consists of G1, S, and G2. And the mitotic phase, or the dividing phase, and that consists of mitosis and cytokinesis. So here's interphase, goes from here all the way around to here, and it's divided into G1, S, which is the DNA synthesis phase, and G2. And then this is the mitotic phase right here, which is divided into mitosis and then cytokinesis. All right, so G1 phase and G2 phase, interphase, called G stands for gap. Okay, so you have gap 1 and gap 2. Gap 2 phase allows the cell to grow large enough and synthesize enough organelles to ensure that the daughter cells will be normal size and functional. Okay, this is the growing phase, gap phase. S is the DNA synthesis phase. Okay, mitosis is not. Well, I just moved all these around and they messed up again. So the S phase, again, is the DNA synthesis phase. This is when you copy your DNA. DNA replication occurs. The DNA is uncondensed and string-like, right? You have to uncondense it, unwrap it. It's this big thing of uncondensed DNA. It's called chromatin. Okay? Now, this is already replicated, so this has twice as much DNA in it as when you start at S phase. So you start S phase here, and you make a complete copy of your DNA by the end of S phase. Right? But it's all uncondensed like this. And if you think about it, when you go through it, if you talked it, let's say if you looked at the amount of DNA in gap 1, and let's say it was 2 nanograms of DNA, if you could weigh it. After S phase, what should you have if everything worked correctly? So in G2, how much DNA should you have if everything worked correctly? If you started with 2 nanograms and you copied everything, you should now have what? 4 nanograms, right? So does that make sense, everybody? If not, let me know. Email me or get in touch with me. All right, let's look at the chromosomes here. Chromosomes, humans have 46 total chromosomes or 23 pairs. You get one in 2 sets. Okay? So one set has 23 chromosomes in it. The other set has 23 chromosomes in it. So total, you have 46. You get 23 from mom, and you get 23 from dad. Okay? So when the chromosomes are copied during S phase, each chromosome is used to make an exact copy of itself. And this is called a karyotype. So right before it goes through mitosis and everything's condensed up, they can do these karyotypes, and they can look at everything and make sure you've got the right number. Everything looks normal. All the copies look the same. There aren't any mess ups here anywhere in here that you can see very easily. So everything's good to go here. Okay. So the cell spends the most of its time in interphase, gap 1. Most cells, a lot of cells like your nerve cells, never even leave it. That's the primary growth phase. It's when your cells are doing their job, whatever their job is. So the cell spends most of its life in this phase. Okay. Okay. Responsible for growth and normal growth and cell function. That's gap 1. That's just a normal cell doing what it does. S phase we talked about, I don't know what happened here, is the synthesis phase. So synthesis of sister chromatids. And like I said, this is where terminology gets a little bit. So you copying of the chromosomes. A chromosome goes from one chromatid to 2 sister chromatids. So here's a centromere. Remember where they're connected. And here are the 2 sister chromatids. Remember, this one chromosome, when it's replicated, turns into this. So now you replicate it. So you've got these 2 sister chromatids. This is one chromatid. This is the other chromatid. They're called sister chromatids. Now, as long as the sister chromatids are connected, this is considered one chromosome. That's what that used to say down there. Okay? This is one chromosome right now. Since they're connected, they're considered one chromosome. Okay? All right, so let's go through the different phases again. Here's G1. Here's your parent cell. You've got, so this is some unknown animal, right? It's only got 4 chromosomes, right? It still has 2 sets, right? But it's only got 2 pair. So it's still diploid because it has 2 sets. All right, now you replicate your DNA. You go from gap 1 to S to G phase. It's still the parent cell, still the parent cell. Now you replicate your DNA. They're connected in the middle. But you still only have how many chromosomes? Over here you have 4 unreplicated chromosomes. Okay? Over here you still have 4 replicated chromosomes. Each of the chromosomes consisting of 2 sister chromatids. But remember, I said they're connected. They're still considered to be one chromosome. Okay? Now you're going to go through the M phase. This is the mitotic phase. This is where you separate the sister chromatids. So at the start of mitosis, the replicated chromosomes condense in those little X's. And then at the end of mitosis, you have 2 daughter cells, right? The sister chromatids have separated. So one's gone there, one's gone there, and gone there. And then over here, this one's gone there, that one's gone over there, that one's gone over there, and that one's gone over here. So during mitosis, the sister chromatids separate, and the 2 daughter cells are formed by cytokinesis. Right? So at the end of mitosis, you have 2 exact copies of the parent cell, 2 daughter cells that are exact copies of the parent cell. So the purpose of mitosis is to distribute the chromosomes to the daughter cells during cell division. It is to separate the sister chromatids and then to separate the chromosomes evenly into the 2 dividing cells. The end result of mitosis, this is a good slide to tell you everything, division of replicated chromosomes and the formation of 2 daughter cells with identical chromosomes. All right, so cell cycle timing again. You've got gap 1, you spend most of your time in gap 1, and then you got synthesis in gap 2, and then you got the mitotic phase, which is then divided into these 4 different sections here, and then cytokinesis. The M phase is the mitotic phase or mitosis. Excuse me, everybody. So the M phase ensures accurate distribution of the chromosomes into the daughter cells of the daughter nuclei during the dividing phase. So that's what mitosis is. It is separating the sister chromatids. You have 4 phases. We'll talk through. Some books divide these into subphases. Some of them combine a couple. It's kind of weird. It just depends on the book. We're just going to stick with the 4 main phases. Okay? You have prophase. So the chromatids condense into those little X's, and the chromosomes become visible. And added to this, the nuclear envelope disappears. Okay? So the nucleus, that envelope around the nucleus disappears. Metaphase, the chromosomes line up across the center of the cell. Then anaphase, the centromeres divide and the chromosomes move to opposite ends of the cell. Okay? And then telophase, the chromosomes begin to uncoil. So they start losing that X shape and get that all diffuse chromatid looking into that loose chromatin. The mitotic spindle breaks down and disintegrates. And the mitotic spindle is the thing that pulls apart the 2 chromosomes, the 2 sister chromatids, and the nucleus reforms and the nuclear envelope reforms and becomes visible. Okay? Now, cytokinesis is actually cleavage furrows form and the cell finishes dividing. So cytokinesis is actually the cell division part. Mitosis is simply separating the sister chromatids, separating the replicated chromosomes. So let's look at it. Here's interphase. Remember, all this nice diffuse, you can't see anything. DNA is uncoiled, and the chromosomes aren't visible even under a microscope. It's all stained blue, but in red, but you can't really tell anything. It's all diffuse in here. Here are your little mitotic, your centromeres with each centriole pair. They're out here. These are things going to connect to it and pull it apart. Okay? And here's that nuclear membrane, right? All right. During prophase, what do we say happens? The chromosomes condense and become visible. You can see them starting to condense here, the purple little things. The nuclear membrane breaks down because you got to be able to connect these little, right, spindle fibers. So the membrane breaks down so the mitotic spindle can attach to the center of the cell. Okay, on either side, attach here and here. And the mitotic spindle attaches to the chromosomes. So you got these little things here, and they're going to attach. So the mitotic spindle is made up of microtubules called spindle fibers. Okay, and the centrosome is a structure that contains a pair of centrioles. Okay? Hold on. Excuse me. All right. So now metaphase. These guys line up single file along the middle of the cell, right? The equator of the cell. You can see them lined up here. And those spindle fibers are attaching to the centromeres, the middle of the little X's, right? So you've got the centrioles here. They're going to attach to the centromeres here with the spindle fibers. So the cell organizes the chromosomes by lining them up in the middle of the mitotic spindle, here in the middle of the cell. And these motor proteins on the kinetochore microtubules, okay, pull each chromosome the opposite direction, causing the chromosomes to line up in the middle of the cell. So they attach to the little centromeres in the middle here on both sides, and they pull back and forth and they pull back and forth until they line up across the middle of the cell, okay, across the equator of the cell, single file. All right. In anaphase, the chromosomes are separated by the spindle fiber, so each sister chromatid goes to the opposite side of the cell. They're like little winches. You know, you've seen a winch on a truck or a tow truck. They start pulling and pulling back and forth until they pop the 2 sister chromatids apart. Okay, the sister chromatids separate, and it starts pulling them in like a little winch. It starts pulling them into the polar sides of the cell. So these divide, and this one goes there, that one goes there, that one goes there, that one goes there. Okay, so the identical sister chromatids are separated carefully to ensure that each new cell will get one. And this is very important. We'll talk about what happens in the next chapter if it doesn't happen correctly. All right. Now telophase, gosh, I hate when you do this, is the reverse of prophase, okay? The chromosomes uncoil They decondense, they become uncondensed or diffuse, and they become invisible. Okay, just a big blob of stuff in there, the blue. You can't actually see the chromosomes, right? The nuclear membrane starts to reform. Here you go. Here's your nuclear membrane starting to reform. You can see it here. The mitotic spindles break down and disintegrate. So the spindles are going to break down, fall apart. And that nucleolus reforms and becomes visible inside of the cell. But you still are only one cell, right? You've just separated the replicated chromosomes, right? So form two independent nuclei. Here's one, here's two. Have formed when mitosis is complete. At the end of mitosis here in this section, the cells have not divided. Mitosis is not actually cell division. Mitosis is separation of the sister chromatids. Now that you've gone through telophase, you'll go through cytokinesis. Okay, you get these cleavage furrows. The membrane starts to pull in on it. You've got all these other cytoskeleton things you talked about, microtubules and stuff, start pulling in the membrane towards the middle. Okay, so you get this cleavage furrow right here, right? And it's going to go all the way around until it goes all the way around, and then the cell pops into two new cells. Okay, watch this thing. If you look at it, it goes round and around, and it pulls down and pinches down until it pops into two new daughter cells. And here's what's going on here. Cytokinesis, cytoplasms have divided, right? Cell division, two daughter cells form. So cytokinesis is when you've separated the cytoplasm, right? And here you can see it going through cytokinesis here. Here's the cleavage furrow here. This one's pretty far along. Here you can see mitochondria moving into the two sides of the daughter cell, right? And here's all the diffuse. You can see the nucleus here, right? Excuse me, everybody. Again, so here you've got interphase. Here's a cell, it's an interphase. You can see the nucleus. You can't really discern any chromosomes. Okay. You've got prophase. You look here, the nuclear envelope's gone, right? Can you see it here? Nope. And you can see the chromosomes starting to condense. Okay. Over here, you've got metaphase. They've lined up across the middle. The spindle fibers have attached. You can even see them. Anaphase, they're starting to pull apart. You can see the chromosomes separating to the poles of the cell. You can see the spindle fibers. You've got telophase, right? Spindle fibers start to disappear. You can't really see the chromosomes. You can just see that diffuse chromatin in there. And the nuclear membrane starts to reform. A little hard to see in this one, but it's there. And then you've got cytokinesis, where the cytoplasms, the cleavage furrows come in, and it becomes two new cells, and then pops into two new daughter cells. Right? So know for the test, draw and describe what is happening in here. So here's a single cell, chromosomes diffuse. Here are your centrioles and your centrosomes. The early spindle fibers start making those apparatus, start making the spindle fibers. So here's prophase, right? The nuclear envelope starts to break apart, and the sister chromatids condense. This is where I said you've got this one that has what they call prometaphase. These two are going to we're just going to make this prophase, okay? The nuclear envelope disappears, right? The kinetochore microtubule starts to connect to the chromosomes, to the kinetochore, right? So we're going to leave this all as prophase. Metaphase, remember, they start pulling on each other until they line up single file across the equator of the cell. That's metaphase. Now, anaphase, remember, they start pulling apart each one, so you're separating the sister chromatids. So half go to this end, half go to that end. Okay. Telophase, the chromosomes start to uncondense again. The nuclear envelope starts to reform. The spindle fibers, spindle apparatus starts to disappear. And then cytokinesis, you start bringing in the cytoplasm. The cell membrane starts to invaginate in. You get these cleavage furrows, and then you have cytoplasm divides, and you have cell division. Two daughter cells are now formed, each with a copy of the original DNA. All right, let's look at control of this a little bit. I don't want to spend a whole lot of time on this, but you remember, this has to be controlled. We'll talk about when that goes out of control, what happens. So the cell cycle length can vary greatly among cell types, and that's what I told you. It's not just 24 hours. And again, it's going to be mostly in variation to G1 phase, right? Some cells never leave G1. So rapidly dividing cells essentially eliminate the G1 cells, okay, the G1 phase. These are cells like the mucous membranes and cells in your intestine and your skin, right? They don't really, you've got some G1 in your epidermal cells, but again, some cells just don't even go through G1. They're dividing so fast, okay? There's also a thing called an arrested state that is called gap 0, okay? So it's called senescence. Some cells will go into G0, which means they can never go through cell division. Okay, it's called senescence. They never divide. They just stay in G0. Okay. So the division rate can vary in response to changing conditions. Okay. Variation in the cell cycle length suggests that cell cycle is regulated, otherwise everything would be the same. All right, so in 1970, researchers fused cells at different stages of the cell cycle, right? So it formed a single cell with two nuclei, and one nuclei often changed phases. So if you had two cells at different stages, right, and you fuse them together, one of those cells' nuclei is going to switch to the other one. Does that make sense? One nuclei often changed phases, okay? So the hypothesis behind this was that cytoplasms in mitotic phase cells have regulatory molecules that induce interphase cells to go into the mitotic phase. And that makes sense. That's regulation and control. And that's why when they put them in here, you had one of the cells that was going to the M phase and the other one that was in G1, those signals would cause that one that was in G1 to go to M phase, right? So injected cytoplasm of M phase frog cells in the cytoplasm of frog arrested in G2 phase, right? In this case. So they got two frogs, right? They inject the cytoplasm of M phase, the cytoplasm. So it's got all those molecules in it. Frog eggs into the cytoplasm of a frog arrested in G2, right? So you got a frog cell that's in G2, stopped in G2. You take the cytoplasm from a frog egg that's going through the M phase, take all the cytoplasm out and inject it into the G2 phase. Those oocysts, those eggs, immediately enter the M phase because all those signaling molecules are there saying you're supposed to be in M phase. So Reacher used the cytoplasm of immature oocysts, right? Injected cells remained in G2 phase. So these are immature oocysts, not in M phase, right? So now it just stayed in G2 because there were none of those signals telling it to go to M phase. And that's their conclusion. The cytoplasm of M phase cells contain a substance that drives immature oocytes into M phase to complete their maturation. Okay? The cytoplasm of G2 interphase cells did not contain these substances. That's why when they put the immature, the G2 into another G2-1, the cells remained in G2. So substances identified as they call them M phase promoting factors or MPFs. All right. So researchers addressed the relationship between MPF and these protein family called cyclins. Now, I don't want to go into a whole bunch of detail in here, but we'll just touch on it. Frog eggs stimulated to divide, cytoplasms then extracted, and the mRNA destroyed, cyclin messenger RNA reintroduced to the extracts, okay, and then samples tested for the presence of cyclin in the MPF activity. So what they're saying here is these research, they look at this relationship. So frog eggs were stimulated to divide, okay, the cytoplasms then extracted, and the messenger RNA for the cyclin, right, this messenger RNA is destroyed. So you're not going to be making any proteins from this. And the cyclin messenger RNA is reintroduced to the extract. So they take it out, get rid of all the messenger RNA so you don't have any messenger RNA to make the proteins. And then you just put back in the cyclin messenger RNA into the extracts, right? And then the samples tested for the presence of cyclin and MPF activity. So MPF increased only after the cyclin protein was translated. So that's what they're saying is here's your control. You take out all the messenger RNA, you destroy it, You add back in cyclin messenger RNA, but it takes a while to make the protein, right? So the MPF, those signal molecules, increased only after those cyclin proteins were translated. So what it's saying here is these cyclin proteins were responsible for regulating that cell division. So the M phase promoting factor, the MPFs, are made up of two distinct polypeptide subunits. Remember, kinase is something that phosphorylates, it adds a phosphate. So a protein kinase is the enzyme that catalyzes the transfer of a phosphate group from ATP to a target protein. It's going to phosphorylate it. And then you've got the cyclin, which is the protein present in different concentrations throughout the cell. Okay, two distinct polypeptide subunits in this, these MPFs. Excuse me, everybody. So concentration of the MPF cyclin increases during interphase. Okay, so it's going to start getting more and more. You're going to build up more and more of this cyclin protein inside of your cells, right? And it peaks at M phase, right, before decreasing again. So all that's saying, there's a little graph here in a minute, is when you're going through G1, S, and G2, the amount of cyclin in the cell starts going up and up and up and up, and it hits M phase and it drops down. The kinase part here, the cyclin-dependent kinase, CDK, active only when bound to the cyclin subunit. Okay, remember you have these two different parts. You have the kinase and you've got the cyclin. So the kinase only functions when it's bound to the cyclin subunit. So you've got those two parts, got here and here. This is going to be the kinase, and it only functions when it's bound to the cyclin. Okay. And the way you control this, you regulate this. Okay. Cyclin is the regulatory protein, right? Cyclin-dependent kinase is the kinase that phosphorylates, right, other proteins to start the M phase. So this together, you have to have these two together. So that this two together, the cyclin-dependent kinase can phosphorylate other proteins to kick the cell into M phase. Now, cyclin-dependent kinase is constant in the cell at high levels. Okay. But remember, you have to have a bunch of these all together for it to function. So until you get enough cyclin, this doesn't do anything. G1, S, and G2, the amount of cyclin starts to increase. And you get more and more of these M phase promoting factors. Now the combined cyclin and cyclin-dependent kinase, you start phosphorylating a bunch of the proteins that are involved in mitosis, and you go through M phase, and then all of a sudden cyclin drops off. Okay, you want to shut that down. And then you start up again. And then you go through mitosis, and it drops off, and you start up again. That's all I want you guys to know about that. All right, these are some of the structures involved. This is a good review slide right here. Chromosome, chromatid, chromatid, they're different. Sister chromatids, centromere, kinetochores, microtubule organizing center. I don't worry about that much, but centrosomes and centrioles, right? A lot of the definitions and stuff that's going on during mitosis. All right, so now we've gone through all of the parts of mitosis. Now we're going to look at different regulation. We looked at how cyclin and cyclin-dependent kinase are the things that regulate cells going into mitosis. But remember, you've got to be very careful on our cells. If you mess up the DNA or something happens, that's bad. If you're lucky, the cell just dies. If you're not lucky, it becomes cancer. So we have these checkpoints throughout the cell cycle, right? And there's a bunch of them. We're going to look at 3 that make sure everything proceeds normally. So regulatory molecules at each checkpoint allow the cell to decide whether to proceed with cell division or not. And if defective, the checkpoint may fail and the cells may start dividing in a controlled fashion. That's if the checkpoint things are defective. Excuse me, I'm sorry, everybody. All right, look at the G1 checkpoint, okay? To pass this checkpoint, the cell's got to be the right size. It's got to be big enough to divide, so the cells are the right size. There must be sufficient nutrients available so the cells can survive. There must be the growth factors must be present. Growth factors are organic factors, epidermal growth factor, VGF, a bunch of these growth factors that make sure the cells divide and grow. Okay, if you don't have enough of those, And you're going to look at the general health of your DNA. You're going to scan your DNA, make sure it's healthy. If you pass all that, you go into S phase and copy your DNA. G2 checkpoint, to pass this checkpoint and go into mitosis, you're going to make sure that that DNA you copied isn't damaged. That's the big one here. Cells copied all of the chromosomes. You copied everything. Not only is it undamaged, did you copy all the chromosomes. If you do not pass this checkpoint here, the DNA is damaged. You didn't copy the chromosomes. The cells stop growing here if the chromosome replication is not proceeded properly, or if the DNA is damaged. Okay. And there are two things that will happen here if it doesn't pass. It can try and fix it. If there's some damage to the DNA, it can try and fix it and then keep going, or the cells will go through what's called apoptosis. And apoptosis is what we call programmed cell death. The cells are like, there's too much damage. We're just going to kill off these two cells. Killing off two cells is nothing, right? You've got trillions of cells in your body, so no big deal. The M phase checkpoint, the final two checkpoints during mitosis, are between metaphase and anaphase. So you ensure that the sister chromatids do not split until all the kinetochores are attached. Because if they separate before you attach the kinetochores, they're not going to separate in the right spot. Okay. Now between anaphase and telophase, this ensures that the chromosomes have fully separated. Okay. So first you make sure they're connected, and then you're going to make sure they fully separate. So the 4-cell cycle checkpoints prevent cell divisions that are damaged or are in G0. Like I said, some of these will just go into senescence. That's another thing they can do where they won't kill off the cell, but they'll be prevented from dividing ever. Then they go into that G0 phase. Again, checkpoint 1, where are we? Checkpoint 1, cell size, nutrients, the right signals, the DNA is undamaged. Checkpoint 2, chromosomes have replicated successfully, DNA is undamaged. Now you activate those MPF, right? So the activated MPFs from the cyclin-dependent kinases together have activated all of those mitotic factors, okay? Checkpoint mitotic pass of all the chromosomes are attached to the spindle fibers and they separate correctly, okay? Mature cells that do not pass this checkpoint can enter this G0, which just means they're prevented from dividing right here. They won't go ahead, they won't copy their DNA, they're just going to stay in G0. All right. Now the important part is you're controlling these, right? You have these genes called tumor suppressor genes, okay? Tumor suppressor genes are genes for proteins that stop cell divisions if the conditions are not, I don't like the word favorable, if the conditions are not right. Okay, one of the big examples is one called P53 we'll look at. Okay, P53 is a quality control agent for that G2 checkpoint. It's the one that scans the newly copied DNA to make sure it is undamaged and been copied correctly. If the DNA is damaged, P53 puts the brakes on the cell cycle and stops the cell in G2 until, like I said, you can fix the damage. Or if it's too significant and you can't fix the damage, it pushes the cells into apoptosis, okay? The problem with this is when this P53 protein is mutated, okay, it allows cells to override that G2 checkpoint and to start to divide uncontrollably, and this is cancer. P53 is mutated in about 50% of the cancers out there. It's one of the most widely mutated, what we call oncogenes, genes that promote cancer growth. It's normally supposed to stop it. So here's your tumor suppressor gene. Here's the tumor suppressor protein, P53. And if it finds damage, it stops cell division. If you get a mutation in it, right, here's a mutation. So now you have a mutated tumor suppressor gene. It doesn't function properly. So the protein fails to stop tumor growth. Right? And here you look at, here's a normal P53. You get the activated P53. Right? DNA is damaged. P53 causes apoptosis. Okay? Abnormal P53, inactive P53 protein, damaged DNA doesn't get detected and you don't shut down cell division. and this can lead to tumor cell formation, proliferation, uncontrolled cell growth. Here they take some of these, they took a viral vector, don't worry about this too much, but what they did is they used the virus to put good copies of p53 back into the tumor cells, and those tumor cells, instead of keeping on growing, they actually would die. So you were slowing down the cancer by fixing p53 in there. All right, so what is cancer? Cancer is out of control or uncontrolled cell division or cell growth. Okay, and it's caused by cells that grow in an uncontrolled fashion, and there's different hallmarks for it. That's part of it. They have to be able to invade neighboring tissues and then spread to other sites in the body. That is cancer. Okay, we'll talk about benign and malignant stuff in a few slides. Cancer develops when genes of a cell involved in those checkpoints aren't functioning. You get mutations in them. You can have p53 I talked about. There's retinoblastoma. You also have DNA repair enzymes that aren't functioning, like BRCA1, BRCA2. If you have those damages, that's when you can start getting cancer. Okay. Additional control mechanisms of cell division fail to stop the uncontrolled growth and the reproduction. Right. The first part is you got to get through those checkpoints with the damaged DNA. So despite their differences, all cancers, again, this is what I'm about, difference between prostate cancer, lung cancer, breast cancer, liver cancer, pancreatic cancer. Despite their differences, all cancers arise from cells in which cell cycle checkpoints have failed. That's the commonality, because those cell cycle checkpoints control cell growth. That has to be messed up for your cancer cells to grow. And here we have some different types in here. Here you've got estimated new cases. This is over 10 years old. I wish they put a new chart in here because some of these have changed a little bit. But in general, you look at it. Prostate cancer, you have a lot of new cases, but you don't have a lot of deaths, right? Prostate cancer, a lot of times, grows really slowly, and it just depends on how fast it's growing in the patient. A lot of people die, males will die later on. They don't even know they have prostate cancer. They'll do an autopsy and they'll find they have prostate cancer, but it was growing so slow they never even knew. We've gotten much better at breast cancer treatment too, so estimated deaths are way down with this one as well. Lung cancer, fortunately, these are old data here, so there's less new cases. The problem is you still have a high mortality rate with lung cancer. Still the leading cause of cancer death. Fortunately, because fewer and fewer people are smoking, using tobacco products, well, smoking would be the only one here. You've seen a major decrease in the number of lung cancer cases. Now we'll see if vaping starts bringing that back up. Colorectal cancer, this is a weird one now because usually this was considered an age cancer. You didn't even start looking at people for it until they were over 50, but in the last couple decades, this is starting to affect younger and younger people, and it's really a big concern. So now they want people to start getting screened at 40 instead of 50. And there's been several famous people who passed away from it lately. Chadwick Boseman, the guy who played Black Panther, he was very young when he died of it. And he actually had stage 4 cancer when he was filming those last 2 movies. I mean, he was amazing that he could do that. Most people can't even get out of bed. Another young actor or comedian just passed away from colon cancer in his low 30s, early 30s. Okay? So this one's one of concern here because of the age. Melanoma, this is skin cancer, the most deadly form. Fortunately, if it's caught early, it's very treatable. Bladder cancer. Non-Hodgkin lymphoma is a blood cancer of the cells once they've left the bone marrow. Okay. Unfortunately, you also have Hodgkin's lymphoma. It's not on here. This is one of the few cancers that has actually been on the increase. Almost all cancers in general have been on the decrease. Okay. Colorectal cancer is an exception, but not as much. But lymphomas have been going up. And lymphomas are one of the cancers that they haven't found a inherited genetic mutation. So it appears to be all environmental, okay, that causes this damage. And you've seen the big ads for Roundup, that's lymphoma. Okay. Kidney cancer cells here, thyroid, okay, very treatable. Endometrial, a lot of these that are caught earlier, very treatable. Leukemias are blood cancers of the stem cells in the bone marrow before they leave the bone. Okay. Leukemias are becoming more and more treatable too. Pancreatic cancer is scary because it is still the least treatable of almost all the cancers. You look at the mortality rate, very high. 45,000 new cases a year, about 43,000 deaths. Okay. I just saw a new little segment. I want to go back and read that they may have found a way to actually start treating these more successfully. Pancreatic cancer has a 5-year survival rate of about 7%. Okay, very scary. It is also the other one that is on the increase is pancreatic cancer. And you've seen a lot more people in the news that died from it. Patrick Swayze, somebody else just had it. Ruth Bader Ginsburg died of it. She had 2 or 3 bouts with it. Very scary cancer. The only lifestyle change they've absolutely linked to pancreatic cancer, interestingly, is smoking. All right, now cells, there's several things that differ from normal cells. Normal cells have these, aside from just not dividing because they're not supposed to, there's several conditions that prevent them from growing out of control. So cells anchor to the dish surface and divide. It's called a tissue culture plate. We grow cells in it, but you have to have a special coating on the bottom that allows the cells to stick to it. If the cells don't stick, they won't grow. It's called anchorage dependence. Okay? So if you don't have that special, a lot of it's collagen, there's some other ones, the cells won't grow. Okay. Now, when the cells have formed what we call a complete single layer or monolayer, okay, they divided, right? Then they're going to stop dividing. It's called density dependent inhibition, okay, or contact inhibition. What that means, when they filled up that area, okay, and they're all touching each other everywhere around, density dependent, they quit growing. That's why your cells, your organs don't keep growing, right? Now, what you can do is you can scoop away some of these cells, and now you don't have that contact inhibition anymore, and the cells will start growing and fill in that little gap until it gets that density-dependent inhibition again, and they stop growing. Okay? Cancer cells differ in several ways. They do not exhibit that anchorage dependence. They start growing up on top of each other, okay, and they'll start floating around a little bit in here, and they don't have that contact inhibition or that density dependent inhibition. Even though this plate is full, they'll keep growing and growing and growing. These are nasty little things, cancer cells. I was working on a cancer cell line, and we changed floors, and one of my little plates got left in the incubator. I went back several months later, and there were cells in there that were still alive, and they should have been dead a long time ago. Very scary. Okay. All right. So properties of cancer cells, again, a tumor forms when one or more cells in a multicellular organism begin to divide uncontrollably. We have 2 different types of tumors. You have benign tumors, which are non-invasive and non-cancerous. Okay. A wart is a benign tumor. Right? Yes, so the cells that started to divide a little uncontrollably, but they don't go anywhere else. And a lot of times they'll stop after a while. There are a lot of benign tumors. Okay? They usually don't cause problems. They can cause problems if they get big enough where they start pushing on other organs in the body, if they're in the inside of the gut, or there's one in the brain called a meningioma. You only have so much room in your brain. If it gets too big, it starts pushing in on stuff. Or they can push in and block blood flow. But that's secondary. They normally don't cause problems. Benign tumors, they're non-invasive and non-cancerous. Malignant tumors are invasive, they're cancerous, and they can spread throughout the body via the blood or the lymphatic system, and then start what we call secondary tumors or metastatic tumors. Okay? That's metastasis. When part of the cancer cells break off, they flow to another part of the body, and they start forming secondary tumors. Cancer cells detach from the original tumor and invade other tissues. Okay? That's the difference. And here you can look at a benign tumor. So this is the benign yellow cells. They grow, but they kind of quit growing. They're not invading this tissue here, are they? They're usually encapsulated. So there's usually they don't go through this basement membrane here. They're not metastasizing. They're not breaking off and getting in the lymphatic tissue or into the bloodstream, right? So they don't spread. Okay? A malignant tumor, you look at it. Okay? Now what is it doing? It's dividing and it's spreading into adjacent tissues. You don't have that little capsule there, right? It's moving into this uncancerous tissue here. It's invading this tissue. Cells are breaking off and getting in the lymphatic system into the bloodstream, and this is just like getting on the highway. And now they can travel to all the different parts of the body and set up those secondary tumors. In fact, most patients don't die from the primary tumor. When they die from cancer, they die from secondary tumors. Okay? And certain cancers like to go to certain places. The bone marrow, the brain, the lungs, the liver is a big one. Okay? In fact, most liver tumors are secondary tumors. So here's a secondary tumor that's set up. So there's a line there. So let's say this is in the liver. All right. One thing about cancer, this thing called the multiple hit model of cancer. Okay. And you have to have more than one, what we call somatic mutation is needed to produce full-fledged cancer cells. There's only one cancer out there where a single mutation can lead to cancer. Other ones take multiple mutations, called the multiple hit model. So here's your chromosomes. Here's a normal cell. You get a mutation in chromosome 2, and it doesn't really do anything. Cell looks about the same. It's still functioning. It's still doing its job. 2 hits, cell starts maybe getting a little bit different. Maybe functions not as well. It's not working quite as well as it should, but it's working well enough. 3 mutations, you start getting some morphology changes. The cell starts looking a little different. Nucleus looks a little weird. Still kind of chugging along, but it's not a malignant cell. It's not going anywhere. It's not dividing uncontrollably yet. And then you get this 4th mutation. This triggers that malignant cell now. And one thing about cancers to remember that is, so you say, well, I've got liver cancer. I got a whole bunch of new liver cells in my liver. Isn't that a good thing? I got more liver. It's not, because cancer cells become undifferentiated. Okay. When cells are fertilized and they start to differentiate into the type of cell they're going to be, let's say a lung cell, okay, when they become cancer, they start undifferentiating. They become unfunctional. So they can't do their job. So you have these unfunctional cells that are growing out of control, and that's the big problem. I'm going to add another slide to this. I want to show you all later. It's not in here. I'll add it and let you know. Okay. So how do you detect cancer, and what are the treatment options for it? And these are getting better and better and better all the time. But the key to cancer is early detection, though. Almost all cancers, even pancreatic cancer, if it can be caught early, there's a chance of curing it. Okay. So here, the advance of technology and early detection of cancers have made it possible to eradicate cancer in up to 50% of cases. That is huge. Just 50 years ago, you got cancer. You probably didn't find it until it was too late. And that's the problem with pancreatic cancer. Another problem is it's usually not detected until it's in late stages, stage 4. And you can go to that when we get to AMP later. But different treatments, you got surgery that just physically removes the cancerous tumors, okay? And they usually follow it by either radiation or chemotherapy. Chemotherapy is chemo's drug. It's the therapeutic drug that kills rapidly dividing cells, right? Radiation treatment is applied directly to the affected area, and it works by disrupting the covalent bonds within the cancer cells. Basically, it shreds the DNA, okay? Or that's what it's supposed to do. The problem is cancer cells are good at fixing that damage. Now, something I want to tell you all here, what they used to do is they would do surgery, and then they would give you chemo radiation after surgery to try and kill any of the cells the surgeon may have missed. What they do now, a lot of times, is they found out they will give you chemo and or radiation before surgery to do what they call shrink the tumor. So if you have a tumor here in the tissue, and here's your tissue right here, right? And as your tumor, what they used to do is they try and cut out the tumor, and they'd give you chemo radiation to try and kill any cells you may have left behind. When they cut out the tumor, they look what they call clear borders or clear margins. They want to make sure they got all the cancer cells. and they give you chemo to try and kill anything they missed. What they do now is get rid of that tumor. What they do now, they found out, is they will give you, so here's the tumor cell, okay, and they'll give you chemo and radiation first to shrink the tumor cell, to shrink the tumor. So now you got this smaller tumor, and here's the area that was where it was, but now you've shrank the tumor, and these should all be dead around here. So now the surgeon can come in and cut out the tumor, and they've got a better chance of getting all the tumor cells. And then sometimes they'll give you radiation and chemo again later. Okay. All right, chemotherapy again consists of what are called cytotoxic chemicals. Okay, cytos, cell, toxic is toxic, and chemicals designed to destroy cancer cells or rapidly dividing cells. Most chemo, that's the problem with it, is non-specific. It goes after rapidly dividing cells. So it prevents replication of cells by inhibiting DNA replication or cell division. The problem is it goes after all rapidly dividing cells, and that's why people lose their hair. They become anemic because your red blood cells are dividing rapidly. They become immunocompromised because your white blood cells are dividing. They have stomach issues because your mucous membranes are dividing. So it's really toxic. Okay, you could cure any cancer with chemo, but you're probably going to kill the patient first. So it's not a targeted therapy. Again, problems also consume normally rapidly dividing cells, white blood cells, red blood cells, hair follicles, cells in your mucous membranes, non-targeted. Radiation uses high energy particles that destroy cancer cells. We talked about that. So it shreds the DNA so they can't keep dividing. It's usually used for cancers close to the surface because that radiation is going to damage any of the good cells it goes through. And again, it used to typically be performed after surgical removal of the tumor, but now they'll do it beforehand sometimes to try and shrink the tumors like I talked about. Now, if a person remains cancer-free after treatment for 5 years, they're said to be in remission. If they are cancer-free after 10 years, that's when they will tentatively use the term cured. A lot of doctors don't like to use the word cured. All right. And now there are a bunch of new targeted therapies, which are getting much, much better. You guys will see much, much better therapies in the future. So they target cancer cells and spare healthy cells. They're making drugs that go after the specific mutations of specific cells. So they'll take the cancer cell, they'll look at it genetically, and they'll figure out where can we target it. Okay? And there are also these things called immunotherapies now. So now they're actually, because your cancer cells, one thing they have to do is hide from the immune system. And so they're getting better and better at retraining the immune system to go after and kill just cancer cells. Okay? So fortunately, treatments will be getting better and better and better. They just had one patient that with one drug, and I forgot how it worked. It was very small. I think it was only 18 people. And it was a rectal cancer. They had 100% response rate for the patients. Tumors were completely gone in every one of the patients. That is unheard of until now. So let's hope it keeps working. All right, everybody, we'll go to meiosis next.